So next we have a talk from Dr. Joe Gray. Dr. Gray is a physicist and engineer by training and holds the position as the Gordon Moore Endowed Chair in the Biomedical Engineering Department at OHSU. His work in biomedics and cancer research have been described in over 400 publications and 72 U.S. patents. To present about measuring better to understand better, please welcome Dr. Joe Gray.
Thank you. It's great to be here and to see all of you here. I'm going to tell you about a long career in science, in particular in measurement science. And just to get things going here, I thought I'd just start with a test question. First, a fact. So one of the things that we know about biology is that each cell in your body contains about 6 meters of DNA. And that's folded up into a ball about 1/10 the width of a human hair. So if you take all of the DNA in all of the cells in your body and stretch it out end to end, how far will it stretch? From Portland to San Francisco, from Portland to the sun, or from Portland beyond the orbit of Pluto? Who thinks shortest distance? Middle distance? Longest distance? There we go. Okay. It will actually stretch 50 times longer than the orbit of Pluto. So how do we know that? The reason that we know that is because teams of scientists have developed tools to help us make the measurements to tell us how big a cell is, tell us how long the DNA in the cell is, and tell us how far it is from here to the orbit of Pluto. The theme of this talk, and actually the career that I want to tell you about, is a career in trying to develop measurement tools. And the concept here is that the better you're able to measure something, the better you're able to understand it. And I tend to focus these days on cancer. And this Newsweek cover that's illustrated on the slide here sort of illustrates what it is that we're about. You can't cure what you don't understand, and you can't understand it if you can't measure it, so we're trying to make measurement tools. Now, I started off my career being fully convinced that I was going to go into nuclear physics. I was a child of the Sputnik era, and everybody was interested in trying to figure out how to make the U.S. orbit a satellite, and so we all got interested in physics. And I got all the way through a PhD in physics. And that was elegant and was fun. But my father developed cancer, and I was stunned to know how little we actually understood about the disease. And so I really wanted to go down the road of asking, could we take some of the measurement principles that I was learning in physics and apply them to cancer? So I'm going to tell you a little bit about that. So when you think about cancer, what are we trying to do? We're really trying to understand a really complex disease. Come back here. Yes. So cancer is actually organized at multiple scales. And if you, this is going to fight with me for a bit, but basically, if you think about life, life starts with DNA, your genes. And those genes encode proteins, and the proteins get organized into cells, and cells get organized into tissues, and tissues get organized into organs, and they make us up. And one of the things that's really remarkable about life is what a size range we have to study in order to be able to understand things about it. So if you're studying humans, then you're thinking about measurement scales of a few inches. If you go all the way down to proteins, you're talking about studying things that are 1/1,000,000th the width of a human hair. And so we need measurement tools that allow us to span this entire range. That's what I'm going to talk to you about. So as we try to understand these complex systems, there are really three stages of discovery or study. The first thing that we have to do is to try to understand what the parts are. The second thing is to try to understand how the parts are assembled. And the third thing is how these assembled parts actually work. And this may be a bit complicated to think about, but one of the important principles that I think that you, if you go into science, are going to have to think about is that we know how to measure the parts, we know how to measure how things are assembled. We don't yet understand how to predict how things work together as systems. And I'll give you an example of that in a bit. That's one of the fundamental challenges of our times. But let me start with just talking about identifying the parts. And one of the reasons for trying to identify the parts is if you think about a disease like cancer, one of the things we want to learn is why does it arise? What's broken that makes cancer happen? And if you have all of the parts and you have a cancer and you have a normal tissue, you can compare them part by part and say, okay, what's broken? Is it the carburetors, the dented fender, and so on? This allows you to focus what it is that you want to study. So we need to have tools to help us break things down into their parts. Now, I entered this field of science a long time ago, and one of the things that we didn't know much about at that time was DNA. What we knew about DNA was that there were about 6 billion base pairs of it. We knew that it was organized into 24 units called chromosomes that sort of condense when cells divide so that the cells can figure out how to separate the DNA. And we knew that DNA was a double helix composed of these bases, A, T, G, and C. But that's about all we knew about DNA. And so I came into the field and wanted to ask the question, what can a physicist do? What can a nuclear physicist do to help us understand DNA? So what I was doing in those days, when you study nuclear physics, the first thing you do is you build one of these particle accelerators, and in essence what a particle accelerator is, a device that takes the nucleus and uses an electric field to accelerate it to really high velocity. You smack it into another nucleus and you look at the parts. And that's how actually you learn the atomic structure of a nucleus. Well, this was just about the time when people had had the concept that maybe you could apply this same strategy to understanding cells. And so what we did was to say, okay, let's take the same principles. Let's take parts of cells, chromosomes, let's say, and squirt them out into air in a liquid jet. It's just that simple. You squirt them out into air, and you vibrate the jet so that it breaks up into droplets. You run the cells through a laser beam. You stain the cells or chromosomes with a fluorescent dye, and you measure how much they fluoresce. And it turns out that if you find cells that are interesting, you can arrange to get them into charged water droplets that fall through an electric field, and they separate. So this allows you to take the cells, the chromosomes, these parts of cells that we were interested in studying, and tell them apart. Now, up until we had gotten into this business, nobody could actually do that. There was just no way to get each of the individual chromosomes out by themselves. We figured out how to do that, and furthermore, we figured out how to take those parts of the cell and put them into bacteria so that they were immortalized and distribute them to the world. Well, that was a nice trick. Who cares? The reason that we care is that it gave us now bits of DNA from each of the chromosomes. And one of the things that we were beginning to learn was that something about DNA. And one of the things about DNA that's really remarkable is that it's this double helix that I think you all know about. The thing that's remarkable about it is that if you heat it up, the DNA strands fall apart. They break in the middle. And if you take some of that DNA that we had sorted and put a fluorescent marker on it and react it with this denatured DNA, it'll bind to it very specifically. And what this allows you to do is to specifically stain these chromosomes. So what this allowed, before we did this, it was very difficult to tell the chromosomes apart. After we did this, it's certainly easy to tell the difference between the red chromosomes and the green chromosomes. It made this possible. That's nice. What's really important about it is that you can also take pieces of chromosomes and stain them, and that allows you to recognize the existence of abnormal genes in cells. This is the basis for a very important molecular diagnostic that's now used worldwide. It's been licensed to Abbott Technologies. And if you were to be so unfortunate as to have a particular kind of cancer, this is the diagnostic that would be applied. So we've taken an idea that originated in physics, and we've applied it to cancer, and done it in a way that allows us to improve how it is that we manage disease. Now, when we were doing all of this, of course, we were keeping notebooks in the laboratory, and I'm sure that you're all being taught that as you're doing your experiments and as you're building your robots and so on, you should keep notes about what you're doing. Right? Okay. So we were keeping notes here as well. And one of the things that happens when you patent something like this that turns out to be important, somebody's going to take you to court over it. So we went to court about this to try to convince the judge that we had indeed invented this. And so they wanted to examine our notebooks. So what did we write on the day that this first worked in the notebook? The only thing that we wrote in the notebook was yabba dabba doo. It's very difficult to actually explain why yabba dabba doo means that the experiment worked. So I'm encouraging you to take good notes while you're doing your work in the future, so you don't have to explain that kind of thing. Okay, that's one way that we've contributed to looking at the parts of cells. Let's talk a little bit about this concept of learning how the parts are assembled. This is an area that's really under active investigation today because of really remarkable advances in microscopy. Microscopy of all types. And the reason that I'm here in Portland, working in the Knight Cancer Institute, is because we have there technologies now that allow us to study living things at all levels of resolution, going from a millionth the width of a human hair up to the size of a human. So this is really exciting because it allows us to begin to learn things about cancer. One of the things prior to beginning work in this area that we sort of thought was that if you had a particular cancer type, it was a rather homogeneous disease. So some of these imaging tools and some clever chemistry have shown us this is a cancer lesion. It's a breast cancer lesion, actually. It's been stained for a particular molecular composition, and you can see that it's far from homogeneous. There's this rim of things around the outside of that cancer that turns out to be really important. We think that that's probably what's keeping the immune system from recognizing that cancer. So we know this because we were able to combine chemistry and imaging to reveal this bit of information. I also showed this example for another reason, and that is that the chemistry that I just showed you there, I didn't explain how it worked, but first off, the use of hydrogen peroxide is an important part of the chemistry. And secondly, all of that staining, all of that work that's really elegant and has informed us about what's going on was started by a summer intern when he was a junior in high school. So we do, in fact, invite students to come into the laboratory and work with us, and they work on real problems. And so if you're really interested in this kind of science, you're invited to get in contact with us, and we'll talk to you about that. So let's talk about something else in the area of microscopy. As you go on in your science career, I think you'll probably all take physics. You can just ignore all of this stuff for a minute, but I think you'll all take physics. And one of the things that they're going to teach you in physics is that if two things are too close together, you can't resolve them in the microscope. And that distance is a quarter of a micron or so. It turns out that if you're a physicist who knows a clever chemist and a good computer scientist, in fact, you can prove that that's not correct. You can resolve almost arbitrarily small things. And I'm not going to go into the details of how this works, but the Nobel Prize in Chemistry in 2014 was given to the three individuals listed here for figuring out this clever combination of physics and computer science and chemistry. So the message here is don't necessarily believe in the dogma. There are many things that you will be taught as you go through school that you will find, if you look closely, may in fact not be strongly supported by science. And so always, as you're thinking about your career in science, be skeptical. Always challenge what you're being taught. Most of the time it'll be right, but sometimes it won't. And you can get a Nobel Prize if you find out some of the areas where they're not right. So that's interesting. So one of the things you'll notice here in this movie that's playing are these little fingers that seem to be coming and going on the surfaces of cells. We couldn't see those in a conventional microscope, but with these new generation super resolution scopes, we call them, we can see them and we can see that they're dynamic. And these are cancer cells that are grown in the laboratory. And what we think you're seeing here are the mechanisms that cancer cells, use to sense what's around them. And once they sense it, they'll retract these protrusions and then invade in that direction. So this is really important because we think that if we can cut off this invasion mechanism, this might have an important therapeutic benefit. Maybe we can just stop them from invading. So this is a laboratory model. We wanted to ask the question, do these things really exist in human cancers? And so that takes me to another device that I wanted to tell you about, and I think this is particularly relevant to this group because this is a robot. This is a slicing and measuring robot. And it is an electron microscope. It's actually made by FEI Corporation, which is located here in Beaverton. And it has two beams in it. One of them is an electron beam that scans the surface of the specimen and gives you an image. And the second one is another beam that allows you to literally slice off 1/100,000th the width of the human hair and scan it again. And you slice and scan and slice and scan. And when you put all of that information together, you get a three-dimensional view of whatever it was that you had in the microscope. And the resolution, again, is about probably 1/10,000th the width of the human hair. So we applied that to a human breast cancer. And I'm going to show you three cells from that cancer, rendered out in this image computationally. The three cancer cells are colored differently. And look at the interface between these cancer cells as we wander into this rendered image. And what you can see here is the remarkable existence of these invasive protrusions that are in these cells. So this is not just something that's going on in laboratory models. This is actually something that's going on in real human cancers, and it's given us some important clues as to how it is that we can think about treating this particular kind of disease. And we also think that this may allow us to tell the difference between lesions that are going to become invasive cancers and those that are not. So a really important bit of information that's come out of this way of thinking. The last thing that I'm going to leave in your plate, because we don't yet know how to do it, is understand the properties of how things interact as a system. If you took a bunch of cars and buses and so on and studied them one at a time, you couldn't know that you could get an incredible traffic jam if you put them all in the wrong place at the wrong time. We need to learn in cancer and in traffic control for that matter, how it is that these complex systems interact with each other so we can figure out how better to control them. And that's important in the context of cancer, because when we think about cancer cells, one of the things you'll know is that cancer cells don't exist by themselves. The cancer cells exist in the body. Cancer cell is next to a cancer cell, it's next to an immune cell, it's next to other components of the body, and it's receiving signals not only from what's right around it, it's also receiving signals from the brain. Turns out that what the brain does has a major influence on how cancers behave. And what we need to understand is this complex interaction dynamic between the cancer cell and its local and its distal environment. This is a computational problem. It's a problem that I think that is probably only tractable with the computers of tomorrow. Until very recently, we have not had the computer horsepower to actually do this kind of study. Now we do. And I think that if you're thinking about a real challenge for the future, this is something that you can take on, and you will have the tools to really make major progress in trying to do this. So it's something that where we really need your help, and it's an area where I think you could make a huge difference. So that brings me to the end of this. I just wanted to leave you with one parting thought, and that is essentially none of what I study today was known or invented at the time I went to school. And so as you think about going forward in your scientific career, don't think about this as something that is going to end when you get your last degree. That's only where it begins. And it's the thing that makes science most fun, is because it's never boring. There's always something new to study. There's always something important to study. And so it's a life of adventure, but it's also a life where you have to be willing, as you heard earlier, you have to be willing to be wrong. You have to be willing to ask stupid questions. You have to be willing to go outside your comfort zone. And if you're willing to do that, you can have a lot of fun in science, and I hope you'll do it. Thank you.
So can you talk about fMRI and that magnetic resonance imaging that is coming along now, and how is that used in?
Well, there's a variety of MRI technologies that are coming along, but the ones that we are deploying are those that are generating signals, if you will, that allow you to distinguish between the molecular components of the cells and to make them sort of distinctly visible in the MRI imaging. We're actually combining that also with PET imaging. So you get a combined image, you get PET and MRI. And so MRI mostly gives you structure, and PET mostly gives you molecular composition. But that's a very powerful anatomic imaging tool. In the back.
So when you were talking about DNA in the beginning, could similar research procedures be applied to RNA?
Yes, so absolutely. So the question was, I talked about how to study DNA. Can you do the same thing with RNA? And the answer is absolutely yes. So there's an entire parallel line of investigation that studies RNA. I didn't talk about that at all, but essentially all of the technology that I talked about there works for RNA. And much of the technology actually works for proteins. So there's this whole field of looking at all of the different types of parts of cells, and that DNA is just one of them. But it's sort of the primary thing that encodes for everything else. Yes?
So there's a push to eradicate cancer by 2020. Do you think that's happening?
There's a push to eradicate cancer by 2020. There was also a push to eradicate cancer by 2015. Do I believe that cancer is going to be eradicated in 2020? Not a chance. I think the concept at Intel was we will be able to get, by 2020, we will be able to take information on cancer and render it into useful information fast enough to be useful for the treatment of that patient. That's what was trying to be accomplished by 2020. Cancer is actually trying to figure out what we're going to do about cancer. That's probably one of the most complicated things that humankind is attempting to do. Because the disease, I didn't have time to talk about it, but if you apply all of these molecular analysis technologies that have been developed to cancers, one of the things that you'll discover is that they're all different. I've never seen two cancers that were identical. They have similar features, but they're fundamentally different one to the next. And so they're different and they evolve quickly. And so we're trying to really control a heterogeneous evolving system. It's really complicated. It's probably as complicated as anything that we're attempting, and it'll take a while to get this done. Okay, thank you very much.